Hi, my name is Mike Mung, and I'm a current undergraduate student at Davidson College. I'm currently a bio major, and I hope to become an orthopedic surgeon in the future. This project was possible due to the NIH-funded SenNet CUSP program. For my project and my time spent in Boston, I researched Raman spectroscopy and imaging senescent cells. So for the background, senescent cells are cells that have halted their ability to proliferate, and a lot of the time senescence protects against uncontrolled cell proliferation, but can also have negative effects on the human body like inflammation and tissue dysfunction. And in the lab at MIT, I researched and learned about Raman spectroscopy, which involves the scattering of light onto molecules to create visualizations and images of the cells. And from the Raman spectra, you're able to create images with its label-free nature, which can be translated to human patients in the future. And IF staining the IF staining that I utilize within my methodology is a technique to utilize the fluorescently labeled antibodies to highlight specific antigens of interest. And so for my research objective, I was able to assist my PI, Dr. Zhan Shu, and my other PI, Dr. Peter So, on their project that was on single cell and label-free identification of senescence by Raman microscopy and by spatial genomics. And my two lab sites were the Shu Lab at MGH and the Spectroscopy Lab at MIT. And so at my time there, my research purpose was to find the correlated images between the IF and Raman. And my objective was not met due to the time constraints, but good progress was still made, and I was still able to educate myself a lot into the field of senescence and Raman microscopy. And so for my materials, I used a cryostat to slice cell tissues, and I used an optimized IF staining protocol for my cell tissue, placenta, and my senescent lung. And I used a standard fluorescent microscope and a custom-built Raman microscope at the spectroscopy lab at MGH. And that shows an image of the particular Raman microscope utilized, and towards the right is the setup and the schematics of the microscope. So feel free to pause if you want to know the specifics of the schematics. Moving on, the methodology I used was to first slice the mice tissue using the cryostat, and then we do Raman imaging for 12 hours, up to 16 hours at times, but it depends on the cell tissue. And then we use IF staining. And then after that, we use the fluorescence imaging. And at the end, we'd analyze the cell tissue and the images, and we compare the bright field, the DAPI images, and the Raman. And so for the bright field, we use the Raman field of view and then the Raman side by side to show the accuracy of the two images. And we use lung tissue 3, 4, and 5 for the senescent cells. And then for all of these tissues, we'd also use the bright field and then DAPI imaging to really show the accuracy of the fluorescent microscope and to show that the tissue was still intact when we utilize Raman. And so for a first cell tissue, which was my placenta, this shows the bright field and DAPI images, and both were utilized by both were captured by the fluorescent microscope, and shows the different staining protocols used. And then for placenta, this is the bright field Raman field of view, and towards the right is the Raman images. Now, it might look a mess, but it shows the unique Raman spectra, and from that you're able to figure out the depth, but also the accuracy and the cellular structure. I also looked at mice brain tissue, and so towards the left is the bright field images, and towards the right is the Doppy images. Now for this one, I had a lot of uh, learning moments as I moved along, and I was given autonomy and independence to make my own mistakes. So the Doppy images did not come out as accurately as the placenta. And I also realized from there that cell tissues are not all made the same. Some are more fragile, which I learned from examining the lung tissue as well. And so this is the mice brain result, and this shows the bright field from the Raman field of view. And unfortunately, I was not able to get any Raman images because the Raman images had unrecognizable features, which was also a learning moment for the future. And from that point on, we usually took the field of view from a corner, usually the right corner, to make sure that we did get accurate images. And so this is our first senescent lung cell, and from the bright field image, it shows a scattering of cells, and this is because I realized too late that the IF protocol was too harsh for a lung cell, which was usually made up of holes and air pockets. And so in the right side shows the Doppy images, which did come out nicely, but none of the cell tissues were intact. Although there was, towards the beginning, this is the Raman field of view, which shows the cell tissue when it was still intact, and then towards the right was the Raman image. And so even though the image is flipped, we're able to see the accuracy and the efficacy of using Raman as a label-free method to image cells. And towards our fourth senescent lung cell, shows the bright field image. And the darker coloration is due to the compression of the cell tissue. And towards the right, the Doppy images did come out nicely. Shows the fluorescent microscope was able to capture these images, but also that the tissue was still intact when we did the Raman and the bright field. And again, this shows the accuracy and also just how effective Raman is in being able to really accurately map out and show detailed images of the senescent cells. And towards our fifth lung, this was towards the later end, so the bright field and the IF came out really nicely because from this point on, I was able to proficiently use the IF staining protocol and also able to gain out my own confidence to really use the fluorescent microscope on my own. And towards the later half, this is the same senescent cell, and we were able to find a good field of view, and it shows the Raman image again, once again, with the unique spectral pattern. It really shows the molecular structure of the cell. And even better is that with the bright field Raman field of view, it doesn't show accurately the depth, but also there's a lot of bubbles from the PBS, which can obscure the viewpoint of researchers looking into the cell. Whereas with Raman, it's label free and is able to show the different wavelengths of the Raman spectra. And so in conclusion, the custom-built Raman microscope at MIT was able to really capture the structure of the mice senescent lung, but also placenta and brain cell tissue there. And Raman was able to be shown as a non-invasive method to image. And I was restricted with my time, but throughout my experience, I was able to really gain the confidence and also the become more proficient and educated, and also really able to fully utilize my training outside of the classroom. And so I hope in the future to determine the correlation between the two modalities and also start analyzing the registration between the IF and the Raman images. And hopefully in the future, I can determine the Raman signatures of senescent cells on my own and without help from my respective PIs. And thank you to the NIH funded SenNet CUSP program for giving me this opportunity, but also to my PI, Dr. Zhen Shu, for letting me operate in his lab as an undergraduate student, and the research technicians Henry Dawson and Junshin Ki or Jill for mentoring me, and much thanks to everybody in the Shu Lab for making me feel welcome, and also to my PI, Dr. Peter So, for letting me operate in his MIT lab, and also Dr. Jun Woon Kang for mentoring me in the spectroscopy lab and at both lab sites, the camaraderie, and for really showing me and inspiring me to continue to do research in the future. Thank you.